In this video we will discuss about fermentation. Fermentation. Fermentation occurs in prokaryotes and eukaryotes. Partial oxidation of respiratory or energy substrate. In fermentation the energy substrate is oxidized and electron acceptor is not an exogenous molecule. In fermentation electron acceptor is an intermediate like pyruvate. Fermentation occurs in anaerobic conditions. But sometimes it may occur in the presence of oxygen. There are two major types of fermentation: alcoholic fermentation and lactic acid fermentation. Alcoholic fermentation. Alcoholic fermentation occurs in yeast and several bacterial species. Pyruvate is converted into ethanol in two steps. In yeast, pyruvate is converted into acetaldehyde by pyruvate decarboxylase. In second step, acetaldehyde is reduced into ethanol by alcohol dehydrogenase. By glycolysis, glucose molecule is changed into 2-pyruvate molecule. In glycolysis 2 ADP and 2 NADH molecules produced. In order to continue producing ADP by glycolysis, pyruvate should not be accumulate. In alcoholic fermentation, pyruvate is converted into acetaldehyde by pyruvate decarboxylase. Acetaldehyde is reduced into ethanol by alcohol dehydrogenase. Lactic acid fermentation. Lactic acid fermentation occurs in muscle cell and in some bacterial species, like lactobacillus. Pyruvate is converted into lactate by the enzyme lactate dehydrogenase, LDH. In animal tissue lactate dehydrogenase found in multiple molecular forms. When different molecular form of an enzyme catalyzes same reaction, such enzymes are known as isozymes. In animals, lactate produced in muscles and transported to the liver, where it is reconverted into pyruvate. By gluconeogenesis, pyruvate converted into glucose in liver. From liver glucose is again transported to muscle cell for glycolysis. This cycle is referred as the Cori cycle.